Thanks, Bahara, for the kind introduction. Yeah, and I'm so excited to learn the experience from both Dr. Cottrell and Dr. Kuma about their experience with the NGS testings of the somatic mosaicism. So in the next 15 minutes, I'd like to share with you our experience and findings in this disease type. And I'm so grateful that both of the presentations have covered the background of the disease and the technical part, so I don't have to spend time on that part, so thank you so much. For this talk, I would like to focus on the genetic spectrum of the RAS variant with the highlight of the in-frame insertions in association with the vascular anomalies. So let's start with a brief overview of the RAS signaling. The RAS MAPK pathway can be activated by a number of the cellular receptors, including the receptor tyrosine kinase, such as the FGFRs, PDGFRs, and the ERBBs, as well as the G protein coupled receptors, as well as the endocrine family members. And these signaling cascade initiate RAS activation by converting RAS from the inactive GTP bound form to the active GTP bound form. And after its activation, the RAS can then interact with the effect proteins to induce downstream signalings, such as the RAF MAPK pathways, PI3K pathways, and the TM1 RAC signaling as well. And then execute a variety of functions that promote cancer development, including the oncogenic transcription, cellular survival, cell growth and metabolism, as well as the cell mortality and migration. As we know that gain of the function missense variants in RAS genes are found in about 25% of the human cancers, with the majority of the variants clustered as one of the three mutational hotspots, including the glycine 12, glycine 13, and glutamine 61. And KRAS is the most commonly mutated and found in about 86% of the RAS mutant cancer cases, and the most commonly seen in lung cancer, pancreatic cancer, and colon cancer. NRAS mutations found in about 11% of the cases and mainly in the melanoma. And HRAS is about 3% of the case and mainly in the bladder cancer, thyroid cancer, as well as the head and neck tumors. So meanwhile, let's go back. Mosaic variants in the RAS genes also have been recently described in the disorders of the somatic mosaicism, including the vascular malformation, nevus formation, somatic overgrowth, and the digital anomalies, and those pictures of the affected tissues that you can try to recall was showed in Dr. Cottrell and Dr. Kuma's presentation. So for us, the genetic spectrum of the RAS variant in this disease type has not been fully evaluated. So we wonder whether the RAS variant in somatic mosaicism is similar to what we have observed in cancer. And to better understand this question, we did a retrospective study using the information and data that collected from the clinical NGS assay targeting this disease type. So a total of 938 cases with suspected somatic mosaicism were submitted to our clinical genomics laboratory in the past 8 years. NGS panel was performed with the average unique on-target coverage depth about 2,000x on the affected tissue, and among those, RAS pathogenic and the likely pathogenic variant were identified in about 60 cases and account for about 6.4% of the total cases, including 36 cases with KRAS, 16 with HRAS, and 8 with NRAS. Here shows the variant distribution of the KRAS in cancer setting based on the COSMIC database, and critical functional domains are indicated with the different colors. As you can see, that glycine 12 and glycine 13 variants account for more than 95% of all the variants, and almost all of them are the missense variants, and indels are extremely rare. Only 2 cases among the dozens of thousands of cases on COSMIC has been shown to be the indels. To compare The lower panel shows that the variant distribution of the KRAS in our cohort of patients with somatic mosaicism. Similar to that in cancer, glycine 12 is the most commonly mutated genes location, and shown in the dark blue in the pie chart. But at the meantime, we also noticed that the in-frame indels account for about 11% of all the KRAS mutated cases and shows in yellow. And this number is much higher than that in the cancer setting. Similarly, here's the data for the HRAS. And the upper panel shows that about 99% of the HRAS variant in cancer are at the 3 hotspot, and there's no well-documented indel variant in the cosmic database. And the lower panel shows that in our cohort of patients with the somatic variant, or with the somatic mosaicism, the HRAS variant at the 3 hotspot account only for 56% of the case. And while the remaining 44% of the case are, as shown in the yellow in the pie chart, actually carry the in-frame insertions, which are extremely rare in cancer. As for NRAS, majority of the variants identified in the somatic mosaicism cohort are at the codon 61, which is consistent with the most commonly mutant in the NRAS variant in cancer. And the in-frame variant in cancer setting is also extremely rare, and only 1 out of the several thousands of cases based on the cosmic database. And in our study, we didn't observe any in-frame insertions in this, the internal somatic mosaicism cohort, probably due to the low sample volume, as we only have 8 cases with the positive NRAS findings. So to answer our question that we asked before, are there any disorders of somatic mosaicism specific RAS variant? And based on our study, that those in-frame insertions are enriched and much more commonly observed in the somatic mosaicism conditions rather than in cancer. And if we take a closer look at those in-frame insertions, we can notice 3 patterns. For pattern 1, we found 4 KRAS in-frame duplications represented by the 4 black lines, and those duplicated regions are about 13 to 33 amino acid long and include almost the entire switch II functional domain. For pattern 2, we found 4 KRAS in-frame duplications represented by these 4 black lines, and those duplicated regions are about 9 to 10 amino acid long and including part of the switch II functional domain. And the last, for the pattern 3, it includes 3 small HRAS insertions represented by the 3 blue arrows. They are about 2 to 4 amino acid long, much shorter than the previous 2 patterns, and located upstream of the switch II domain. So the next step, we would like to know whether there are any specific phenotypes associated with these in-frame insertions compared with the hotspot variant. And to answer this question, we went over the clinical indication and the possible phenotypes collected at the clinical testing for all the 60 cases with the RAS variant, including 49 cases with the missense variant and 11 cases with the in-frame insertions as we described in the past couple slides. And interestingly, we found that all the 11 individuals with the in-frame insertions were tested with the vascular malformation or similar conditions, while the rest, the hotspot missense variants are associated with a broader clinical presentation beyond the vascular malformations, and some of them including nevus, somatic overgrowth, digital anomalies, and multi-system findings. So this observation suggests that the in-frame insertions associated with the vascular anomalies and really with the additional findings. And we also analyzed the age distribution as well as the variant allele fractions in those cases. And the median age at testing for individuals with the in-frame insertion is about 12.3-year-old, while the median age with the hotspot was about 8.6-year-old. And this finding is consistent with the milder phenotype spectrum associated with the in-frame insertion variant. And the variant allele fraction observed in case with in-frame insertions about 1.4% at the median, and the median VAF in the hotspot missense variant is about 8.2%. So the reason and clinical significance of the relative low VAF associated with the in-frame variant is not very clear now, but there are several hypotheses that I would like to share with you, and we can start with the discussion as well. So first, in general, the indel VAF, NGS calling is more challenging than that the single nucleotide change. So it's possible that those VAF of the in-frame insertions are actually underestimated based on the NGS caller. And second, it's possible that the RAS activation by the in-frame variant is not as strong as the hotspot variant, and there are some of the functional studies using the animal model kind of like support that hypothesis. So the cell replication is slower, and less affected cells present at the time of testing. And third, as almost all the cases with the in-frame variant has associated with the vascular anomaly as the sole clinical presentation. So it depends on the biopsy method and the location. The tissue biopsy for testing may only contain a small portion of the affected tissue, and with extra surrounding normal tissues, which can cause the underestimated variant allele fraction in those results. So the lower VAF, the variant allele fraction of the in-frame insertions observed in this study, further demonstrate that the clinical utility of the deep sequencing in diagnosis of the somatic mosaicism conditions. In summary, although most gain-of-function RAS variant clustered at the 3 hotspot codons, 12, 13, 61, we identified a group of the in-frame insertions observed more frequently in the disorder of the somatic mosaicism, and including the 3 patterns that we described in this presentation. And those individuals with RAS in-frame insertions are associated with the vascular malformation tends to have elder age at the testing and the lower variant allele fractions compared to the ones with the well-established hotspot. And with that, I'd like to thank all my colleagues at WashU in this clinical genomics laboratory, and especially Claire Ho, who is the former LGG fellow, did most of the work of this study. I'd also like to thank all the collaborating clinicians and the patients and their families who contributed to this work. And thank you so much for your attention, and happy to take any questions.